I will get started now. My name is Sarah Kennedy. I'm the founder and CEO of Calikerb. I'm delighted to introduce Dr. Edward Walker. Dr. Ed has spent the last 14 years, he told me his hair wasn't grey before this, and he works for Plant and Food Research, the largest government research institute in New Zealand with over 700 scientists. Dr. Edward Walker received his PhD from the Auckland University in berry fruits and antioxidants, and has continued to work in plant-based research. So I'm going to hand over to Dr. Ed to take you through the last 14 years.
All right, hey everyone, can you hear me okay? Yep. All right, so I'm going to be talking today about the development of a GLP-1 stimulating nutraceutical. And really it's less about the fact it's a GLP-1 stimulating nutraceutical, and it's more about what the process you should take if you want to develop a scientifically validated nutraceutical product. Because as we're all aware, there's a lot of stuff in the market, and there's a lot of stuff that doesn't really work. And so it's more about the journey that you go along, what the research you should do in order to make sure your product really works. So this is the last 14 years of my life in a few slides. So to start with, there's an accreditation statement. So I don't know what this means, but for you guys, you can read through it. And a disclosure. So I have received research grants from Calikerb, and that started around 2018. So the first 8 years of this research were done purely government funded, and then there was joint research. All right. So I am an employee at Plant and Food Research. So as Sarah said, it's the largest New Zealand government-owned research institute. So it was founded in 2008, but essentially the history dates back to around 1926 when the initial sort of government research organisations were funded. So in New Zealand we have probably a little bit different from in America. Most of the research is government funded, so we only have a very small amount of industry funded research. So the government funds pretty much everything. Plant and Food Research does essentially plant research. So we look at things like development of new cultivars. So for all those people who know about gold kiwi fruit, so the original gold kiwi fruit came from Plant and Food Research. Things like fish breeding and consumer, like what fruit tastes good, what traits do you want. But the area that I'm involved in is really the health and nutrition space. And so we've been developing novel plant-based treatments for health for a long time. And Amarasate, which is the GLP-1 stimulating extract, is really the most recent of them. We've got more than 700 research staff, over 14 sites, and we've got representatives in mainly New Zealand, but also some in America and in Australia. So here are our locations. So basically all through New Zealand. So you have up in the North Island, we've got potato growing in Pukekohe. Going down through the South Island, the hops are grown in the top of the South Island in Nelson and Motueka, which is exceedingly beautiful. I recommend you visit. And down south in Clyde and Gore, we do basically pip fruits. And then all the way through the midsection, we've got kiwi fruit growing. Then a couple of branches in Australia and one in Oregon in the US. All right. So what I'm talking about today is really stemmed in a government-funded program called Foods for Appetite Control. Now this was a $20 million New Zealand 6-year research program, so that's around sort of $15 million, $14 million US research program. It was led by Dr. Kevin Sutton, and the goal was to develop a gut-targeted plant-based nutraceutical for appetite control. We wanted it to basically be able to empower people to make healthy food choices so that they, when they're on a program, they can control their appetite and make the food choices they want. And we wanted it to be non-diet specific. So everyone's got different favorite diets. So personally, I'm a sort of a low-fat, whole food kind of guy, but some people are ketogenic, some people are Mediterranean. So whatever diet you choose works, it needs to be compatible with that. And to be honest, the key factor in diets is really compliance. So if you put someone on a diet, if they can stick to it, then they're going to have a good chance of success. So we really wanted it to help with all diet types. So we got that initial government funding for 6 years, and then post that there was sort of commercial, well, government funding for another 2 years, and then commercial government funding since 2018, sort of co-funding's been going forward. All right, so the big question is, why did the New Zealand government in 2010 decide it wanted to fund a $20 million nutraceutical research program? And I mean, it's a problem we're all familiar with. It is the global obesity epidemic. So these are sort of updated stats, but you're probably all aware of them. So we're looking at more than half of the world's population being obese by 2035. And this is, a big change from what's happened previously, where most of the obesity has been in the Western world, which is arguably more able to cope with the obesity epidemic and the diabetes, the heart disease, and the risk factors that are associated with it, such as increases in certain forms of cancer. We're also looking at obesity being the #1 contributor to ill health, so more than infectious disease. So we're going from an infectious disease demographic where people are ill to a lifestyle disease demographic. In New Zealand, so we're pretty similar to America. So the latest figures, 1 in 3 people are obese, and about another third are overweight as well. But the biggest and most concerning trend we have is the increase in obesity in adolescents and children. So we're looking at New Zealand in 2020, we had 12% of the population of 2 to 14-year-olds is obese, and that was up 3% on the previous year, so it's tracking right up. And of course, if you're overweight and obese as an adolescent, the chance of that being true as an adult is huge. So it's a major predictor for overweight and obesity when you're older. So obesity is set to be almost 3% of GDP as a cost, and so to put that in context, that's like what COVID cost the world economy. So it is absolutely humongous. And it's important to realize that we're not just talking about ill health as far as physical well-being goes, but there's so many emotional and mental health aspects to obesity as well. And just that note, obesity is a long-term chronic disease that needs long-term treatment. And I think the attitude that you can just get someone to lose weight and it's fixed is not true. You need to manage them, they need to have a lifestyle change, it needs to be for the rest of their life. All right, so why didn't the government just say, let's just do the status quo and do diet and exercise? So that's been what's been suggested for, well, really thousands of years. So if you go back to ancient Greece, they would use diet and exercise. They'd prescribe you to go for a walk, and they'd also prescribe diet modification. They'd even give you emetics so that you would vomit your food up, right? So they'd modify food intake. So we've done this for thousands of years, and the success is really mixed. So you will get people who are highly compliant with the diet, and you give them the information and they follow it and they're able to follow it, and you get good success. But the reality is that's not most people. And if you look at long-term weight loss from diet, it varies between studies, but 20% of people, if they're going on a diet and they don't have a lot of support, are going to get long-term some weight loss. Why is that? Well, the reason is because people cheat because they're hungry. So the #1 issue with people cheating on diets is they're hungry and they're craving certain foods, and so they can't stick to the foods prescribed on the diet. So we really wanted to address the hunger issue. So essentially, appetite control was the key to making people compliant with diets, or at least probably the major component. So what about the medication? Well, in 2010, when we started this program, there weren't a lot of good medications available. So there were a couple that had recently been pulled from the market, so one of which you never got in America, which is it's quite a cool one actually. So if you look at rimonabant, so that is an inverse agonist of the cannabinoid CB1 receptor. So basically think anti-marijuana. So it's quite popular in Europe. And so the idea was that if you smoke marijuana, you get the munchies. So this drug would block the receptor marijuana targets, and so it would decrease your appetite. So the question is, what else does marijuana do? So we got anyone in the audience who can fill us in? No? No? It was really different in California. There were lots of people. So it makes you high, right? So maybe somewhat unsurprisingly, this medication led to people becoming depressed and suicidal. So they pulled it off the market in Europe in 2008. So it's only 2 years it was on the market, and then they realized whoopsie, we've made a mistake. And then you've got sibutramine, which is one of these, I mean, they call it, I think it's Reductil is the product name, but it really hypes you up, so it's a stimulant. Mode of action is monoamine uptake, so neurotransmitter reuptake inhibition, so primarily looking at your noradrenaline or norepinephrine, and that ends up activating some of your beta-adrenergic receptors and increasing your energy expenditure. Now again, somewhat unsurprisingly, and if you asked, a bunch of scientists back when this product was launched what the side effects would be, you'd say, well, anyone who's got cardiovascular stress is probably going to end up having issues on this. What is obesity linked with? Cardiovascular problems. So what happened? They found an increased rate of cardiovascular events for people on the product, and it was withdrawn from numerous countries, including New Zealand and I believe America as well in 2010. So just because it's topical and this talk is about GLP-1, we have a GLP-1 stimulating or a GLP-1 receptor activating peptide here. So this is semaglutide, so the Wegovys. And what it is it's a synthetic version of a gut hormone called GLP-1. So every time you have a meal, your gut sends a signal to your brain telling you you're full. And that signal is primarily three hormones. One is called CCK, which is short-term, quick-acting. One is GLP-1, which is a longer-term, sort of moderate, medium-term acting, sort of appetite-suppressing signal. And the final one's PYY, which is a sort of a less potent but even longer acting. And so they decided, the drug companies, to make a version of GLP-1 which they could use as a drug. Now you can't use GLP-1 as itself because it's only got a half-life of 2 minutes. So in your body, you want that appetite-suppressing signal to go to your brain, tell you you're full when you've got food, and then to turn off when you're not hungry anymore, when you're not full anymore. So they took GLP-1 and they modified it at a little point here so that it was not cleavable by an enzyme in your body that breaks it down. And so that turned the half-life of GLP-1 from 2 minutes to 1 week in the case of semaglutides. And so that meant that instead of having, when you take an injection, instead of having GLP-1 activity which goes up with a meal and goes down, you have GLP-1 activity that is high. It's high in the morning, it's high in the night, it's high in the evening, it's high when it's normally 0 when you're asleep, and it's just constantly elevated. And that's really great for suppression of hunger, but it's probably not great for mimicking your natural biological patterns of GLP-1 activation. So what are the challenges with semaglutides? Well, really it's the high rate of non-serious side effects. So we see a very high rate of things like gastric upsets, diarrhea, etc. So a lot of digestive issues, and occasionally more serious side effects such as gastroparesis, but they appear to be quite uncommon. There's also the high cost of use. So it's expensive, you have to inject it. Most people take it for a year, and to be honest, the people who are most in need are the ones who can't afford it. So it's out of reach for a lot of the most needy people. So what about nutraceuticals? So again, there were not a lot of nutraceutical options available. So nutraceuticals, in theory, are this wonderful sort of merging between nutrition and pharmacology, where you take the best of both, and then you have this wonderful product in the middle. The term was coined by Dr. De Vlies in the 1980s. So the founder of the nutraceutical science. In the 2000s, he actually said that it had failed. Nutraceuticals had not lived up to their promise at all. And then in about 2020, he came back and said, well, they can if you do the research properly. So that's really where we are with nutraceuticals. It's about doing the research properly, because what tends to happen is you tend to get something which takes the worst from both. So you end up with something which takes the inefficiency from food and then takes a bunch of side effects from pharmacology, and then you've got a product which is really no good. What products that actually worked? Well, there was ephedra, which if you're old enough you'll remember, which is basically it's a Chinese herb, and it contains a whole bunch of compounds which activate. They're essentially really similar to adrenaline or norepinephrine, and they activate the beta-adrenergic receptors. So again, it hypes you up, it burns fat, but as you'd probably expect, it has a lot of side effects. The FDA banned this product in 2004 because it was 1% of the herbal sales and 94% of the herb-related reports to the poison control centres. So, does it work? The answer is yes. Is it safe? Well, probably not. So what we decided to do was we decided to take, our approach was gut targeting bitter plant compounds. So we're getting plants that are bitter plant compounds targeting to the gut. So why did we do that? Why do we think this would work? Well, plants have had a huge history of modulating appetite in people. So all you need to do is think about an apple tree and an apple. Right? So what does that apple tree want you to do? It wants you to eat the apple, it doesn't want you to eat the seeds, and it doesn't want you to eat the leaves around it. So what the apple will do is it will make the flesh of the apple delicious, and then it will make the seeds taste bad, and the leaves around it taste bad as well. So, and as part of that, it makes them, one, bitter, because bitterness is not something that humans like to taste, and two, it also will put in appetite-suppressing compounds. So if you look at apple leaves, there's actually a mild appetite suppressant in the apple leaves that will also regulate blood glucose levels. So plants are sitting there, they're going, look, I'm going to change my physiology so that I can trick these mammals into not eating the bits of me that I don't want to. And so they've evolved this system, essentially, to do that. You can see it's evident in animals like cows, where if you increase the load of bitterness present in their food, the cows will actually eat less. So you give them a whole bunch of feed, and you make that feed bitter, they eat less. If you make it less bitter, they eat more. Right? So it's really this triggering of this bitter taste as a potential marker of something is not good to eat, even though the plants are just tricking us. So the real question here for us was, well, is this bitterness something we can look at, and can we use it as a way to regulate appetite? So the second, I guess, bit of evidence we had that bitterness might have been interesting was that it's been used a lot historically. So if you look at sort of various cultures, you can show both positive and negative regulation of appetite using bitter foods. So in Rome, they'd use a bitter-infused wine, which they'd drink just before a meal so that they could have like a huge feast and stimulate their gastric juices and their hunger. And then there are a couple of examples of suppression of appetite. So in the Highlands of Scotland, they'd use a bitter heath pea to suppress appetite during times of famine. Now this is actually reported to be used in the court of King Charles II as a treatment for obesity. They use this bitter heath pea to try suppress their hunger. And there's also, or even currently, in the Kalahari, Bushmen will use a bitter cactus to suppress their hunger and suppress their thirst over on hunting trips. So there's a lot of really interesting information around bitterness use historically, and the same is true for Chinese medicine and Vedic medicine as well. Now, this proposes an interesting question, because if this was just on your tongue, surely it would all be negative. So if you were just tasting it on your tongue, why did the Romans get a stimulation of appetite? Well, we got an answer to that in about 2011 when a study was published in mice where they gavaged a whole bunch of really nasty bitter synthetic compounds directly into the stomach of the mice. And what they were able to show was that acutely you had the stimulation of appetite. So essentially in the first sort of hour or half an hour or an hour, they actually ate more food. So imagine what the Romans were doing. They were taking that bitter-infused wine immediately before a meal to stimulate their appetite. But then chronically, there was a really big suppression of appetite. So you can see over the course of sort of 1.5 to 4 hours, the mice were eating substantially less food. So there seems to be this, a temporal differential between it. So when it's taken really quickly before a meal, about when it hits the stomach, stimulates appetite, about when it hits the small intestine, suppresses appetite. Now this led us to form a concept called the bitter brake on appetite. So this was the concept based around the whole project. And the idea was that if you send bitterness to the stomach, you trigger a pro-appetite signal. The idea is that you've probably accidentally eaten it, you didn't mean to, and so your stomach sends a pro-appetite signal to your brain saying, look, you're not going to eat that again because it tasted bad. Go eat something out, dilute out whatever's in your stomach, seal your stomach off so it digests more, it'll get the gastric juices going, right? So dilute it down and try to destroy it. Now if it's in the small intestine, then it's getting absorbed. So whatever has been done before isn't working. So the only option is to send a signal to the brain to say, look, just stop eating. It's probably not tasting bitter on your tongue. So the only option is just don't eat it. And also to send a signal to the stomach that says, don't empty. So delay gastric emptying, because whatever's in there, we want to give it the best chance possible to break down. Now this lines up really, really well with gastric hormones or intestinal hormones. So if you look at the hormones in the stomach, you have a hormone called ghrelin, and this is a pro-appetite hormone that will stimulate hunger. And then as you go further down the intestinal tract, you get CCK, which again is this anti-appetite hormone. It's very potent, but it has quite strong rebound effects. So it suppresses appetite like that, but after about an hour or so, you actually end up eating more. So in isolation, it's not really very good, but acutely, it's very powerful. Then you have GLP-1, which is a longer term. It's spread out through the lower parts of your gastrointestinal tract, and it's a longer term appetite suppressant. And again, right at the bottom, you have PYY, which is again a sort of quite a gentle appetite suppressant, but longer term again. So these lined up really well with that pro and anti-appetite effect of bitterness on the gut, where ghrelin is in the stomach, so if you trigger that, you're going to get pro-appetite, and CCK, GLP-1, and PYY are in the lower intestine. So if you trigger them, you get an anti-appetite effect. So how do we make a nutraceutical with this information? Well, we asked ourselves 3 questions. The first one was, are there actually bitter taste receptors in the gut? So is this real? Can we actually basically have bitter taste buds? Because no one knew. Can we activate this system in the lab? So if we get cells from the gut that have bitter taste buds, can we trigger these with plant compounds? And 3, can we demonstrate this clinically? So can we show this works in people? So the first study we did was asking, are there bitter taste buds in the gut or bitter taste receptors? And so what we did was I went into hospital and asked people to volunteer bits of their intestine. So this took a long time, but essentially what we were looking for was parts of the intestine, so 5 parts from the top of the intestine, 5 parts from the bottom. So if they were having a gastroscopy, getting 3 bits of the stomach and then 2 parts of the small intestine, and if there was a colonoscopy, asking for 3 parts of the colon, the rectum, and the end of the small intestine. Right? And the inclusion-exclusion criteria was really tricky on this. So we basically wanted people who were going into hospital to have a procedure, so a gastroscopy or a colonoscopy, who didn't have any gut problems, no diagnosed gut problems, and didn't have any gut problems diagnosed at the end. So they were essentially people who had unexplained pain or unexplained bleeding, and then didn't get diagnosed with anything. Right? And so that became quite tricky because the people who'd had lots of gut diseases were like, oh yeah, take some biopsy samples, don't worry about it, because they've had hundreds done before. But the people who had no health issues were really not keen to give them. So it took about 2 years to do this research. We did analysis using quite a cool thing called nanostring. So in the human body, there's 25 bitter taste receptors. So you've got you've sort of got 1 sweet taste receptor, you've got 25 for bitterness. We don't know what a lot of them do, and they're quite hard to detect. So we use the technology which basically binds to the RNA and then lies with a sort of a fluorescent coloured probe, and then lies flat, and you can just count the number of sort of coloured combinations. That was the technology we used, and it was really sensitive, so it let us detect small amounts of these taste receptors. And what we were able to do is we got 30 participants, 15 to give samples from the top of the gut, 15 from the bottom of the gut. We had 28 total because 2 people gave samples from both, so they were real soldiers. And we could show essentially that there were bitter taste receptors all the way through the gut. So they were there, so that was the first take, right? So we had that information, and they were somewhat elevated in the gastric fundus and the terminal ileum, and that makes sense because the gastric fundus is where basically the fluid sits when you've had food in the stomach, so anything floating on it would be detected there, and the terminal ileum was where you'd have potential bacterial overgrowth from the gut. So next one is, can we activate the bitter brake in the lab? So this was going to be our sort of in vitro lab-based experiment, saying, if we take one of these gut cells which has bitter taste receptors and these anti-appetite hormones present, can we use plant compounds to activate it? And so what we did here was we got these, what they call enterendocrine cells. So these are the cells from the gut that have the bitter taste receptors and the appetite suppressing hormones, and we did high throughput screening. So we ran around 1,000 plant extracts and pharmaceutical compounds through to see what could trigger the release of appetite suppressing hormones. And we looked at it in 2 ways, and this is quite important because with these cells, you have a cell and then it has a little packet, a little vacuole of these hormones in it. So you can make them release those hormones by killing the cell. So I can just put on anything. I can put on detergent and I can release those compounds. So if I want to do a paper that says X extract releases these hormones, all I have to do is put on enough for it to be toxic to the cell. So what you need to see is you need to see that signaling pathway. You need to see the internal calcium change, which means you've triggered the receptor, and then you need to see the hormone release as well, because if you only got one, then you don't know your results are real. And so we did that for all of these, like 1,000 different compounds and plant extracts. To be honest, not much worked. And that's somewhat not surprising, because if the system worked really, really efficiently, then we'd probably know about it. You'd probably be like, oh, I've had some coffee, I can't eat anything, because coffee's a bit bitter. So it was really resistant. We only got maybe 4 hits out of the entire 1,000 compounds, and only one of them was really suitable to give to people. And so that ended up being a hop extract, so a hop like you use to make beer. So obviously beer is very bitter. Now these are not, strictly speaking, the compounds you get in beer, because you boil beer and it changes them a little bit. These are the ones that are actually in the hop flour. But what we were able to show with these hop extracts is that you could get a dose response, so that's really important. So as you increase your dose of the hop extract, you get more calcium signaling, so that's that internal signaling, so you know you've triggered the bitter taste receptor, and also we get increasing in hormone release. So in this case it was CCK we measured, but GLP-1 is identical because they're both present in that vacuole. We also could show that there was differences between the extracts. So on the top figure there, the red line is the hop extract we ended up using, and it's quite a potent, quite a steep curve, and the blue line below it is a standard hop extract, which is not as strong. Right? So next, we also wanted to say, what's the bioactive? Because it's all right having an extract, but if you don't know what your bioactive compound is, you can't standardize it, you can't make sure it works from year to year. And if it turns out you had a particular year where you grew a plant and it made a compound that it doesn't usually make because of weather conditions or exposure to pesticides or something, then you're out of luck. So what we did was we did HPLC, so high performance liquid chromatography testing, and we could show there were pretty much 6 compounds present, and these were the alpha and beta acids from hops. So these are the primary and secondary bittering agents in hops. And so those are those 3, and at the top. And then when you ran them, so when we ran them as individual as we could get them, so they're quite hard to separate because they're very similar compounds, but we separated them the best we could, and we tested them as individually as we could. Some of them are slightly mixed, and what we could find was that the most potent compound was likely to be cohumulone because it had the lower CC50, so that's the sort of off yellow line which comes up before the other ones, but all of them had some activity. So all of the alpha and beta acids were somewhat active, and that makes sense because they're somewhat bitter. Cohumulone being the most bitter, most orally bitter, and being the most bitter in the assay makes sense as well. So that was quite good. We then wanted to say, okay, we got all this data from the biopsy study on bitter taste receptors. Are the bitter taste receptors that cohumulone and hops activate present? And the answer is yes, they are. So if you look at the top line, that is TAS2R14, so that's triggered by the beta acids, so they're the less bitter of the hops compounds, but there's quite a lot of the receptor. Then TAS2R40, which is triggered by cohumulone and the other alpha acids, is also present. And then interestingly, TAS2R1, and this is what's triggered by the bitter compounds present in beer, is not present. So that's in the margin of error for the assay. So that explains why you can't drink beer and lose weight, because you don't have the receptor required to get that gut signaling to beer. It's on your tongue, but it's not in your gut. All right. And then lastly is for the preclinical work, we did a gut dispersion assay. So this extract's like a wax. It's like a thick wax. And as all good scientists do, we put it in a pill and took it ourselves, even though you're not meant to. And what we found is that occasionally it would pass through you intact. And I won't tell you how we figured that out. But so it was clearly not dispersing in the gut. Now when you have a gut target, what you want is you want it to go to your point of bioactivity. So for us, that's the upper small intestine, and disperse a bit, but not so much that it gets to a low concentration. You don't just want it to go and become really dispersed, because then it won't be potent or concentrated enough to activate. So we added a small amount of oil and ran it through simulated digestions. So these digestions are essentially a fluid a fluid which mimics the stomach or the small intestine, and it's on a shaker. And what we could show is if you didn't add any excipient, or no oil in this case, it just ended up being a glob, like we experienced when we took it. And if you added a small amount of oil, you could make it disperse a little bit. So that helped with the formulation. So we had a formulation to use clinically. So next, can we demonstrate clinical efficiency or efficacy? And so this was really about saying, okay, can we trigger the bitter break? Can we trigger these hormones to go to the brain in people? So this is a study design, and it's a very sort of fixed, we're in a clinical trial design, and what it involved was cannulating. So we got 20 guys, and we cannulated them, and then we gave them a fixed breakfast. So that's a breakfast which is 2 megajoules, and it was fixed energy, just to sort of set off their hunger profiles and their hormone profiles, so they're all standardized. We then gave them the hop extract targeted to either the duodenum, so that's the D, or towards gastric release. So the D was in a delayed release capsule and the G was in just a standard capsule before an eat till you're full lunch. So that's basically it was a pasture and meat meal, and they just ate it until they were comfortably full. And then we had an eat till you're full snack, which was ham sandwiches. Cannula was removed at about 5 o'clock in the evening, and all along we took samples. So where there's a B, we took a blood sample, and when there's a V, we asked them how hungry they were feeling. Now this design was primarily to show changes in food intake and changes in hormone levels, so there wasn't really a lot of changes in the VAS data, but for the important stuff, we got some really interesting results. So firstly, what happened with ghrelin? So this is this hormone in the stomach which stimulates appetite. Now we were hoping that we would get an effect in the gastric targeted pill and not in the duodenal targeted pill. What's happened is we've actually got it in both. And the reason for this is probably twofold. One is you do have some gastric expressing cells in your upper duodenum, so we're always going to get a little bit of stimulation, but you also have quite a bit of, about up to 10% of your duodenal fluid actually mixes back into your gastric compartment, which means that there was always going to be the chance of stimulation of the gastric hormones. So we basically showed that you could get, if you looked just before the ad libitum lunch, an increase in both of the treatment groups. So that's what the Romans were seeing. So we were hoping to avoid that, but we didn't quite manage to. But nevertheless, that's a positive result because it shows we're getting bitterness that's effective into the gut. And if you look at the placebo, which is the white line, you just get a small increase in ghrelin leading up to the lunch, which is exactly what you expect. Okay, what about the appetite suppressing hormones? So CCK, again, we get a small increase of CCK to the fixed breakfast. It's what you expect, goes down and then it shoots up after the lunch. Again, just how you expect the placebo, which is the bottom white line. So it goes up, goes down, goes up to the snack. That fold change is what you'd expect. And then both the gastric and the duodenal treatments were able to stimulate that significantly and really quite a lot over baseline. So this is a big change for people. What we did see is the gastric treatment was slightly more potent acutely and then started to die off chronically. So as it went longer, it was a little bit less effective. And this again is what you'd expect because you're getting the gastric release is going to be more constant, well, it's going to be more complete at the top of the gut or just after the stomach. But because it's more dilute, it's going to dilute out below your critical sort of threshold of activation as you go down the gut. What about GLP-1? So this is the one everyone's interested in. So GLP-1 was an interesting one. So what we saw here was we saw again the same kind of profile of a little bit of response in all of them to the breakfast before the treatment. So that's good, they're all the same. And then you get an increased response with both the treatment groups, both the gastric and duodenal targeted pills. But again, the duodenal targeted pill, and you can see it really particularly towards the end of the after the ad libitum snack is quite a lot higher. So it's got this lastability that the gastric treatment doesn't. Now these have got quite big error bars, and what we had was one individual who had a humongous baseline GLP-1, so some sort of interfering factor. So he basically had 40 times the average of everyone else, which causes a big error. If you normalize to baseline, which is the small figure in the bottom corner, that's what the data looks like. It looks sort of essentially the same, just less error bars, and it more highlights that the duodenal treatment was more effective as you go longer in the time frame, so towards the end of the study day. Okay, and what about PYY? So again, PYY, the same individual had an interfering factor, so we ended up with with more error bars. PYY is a bit harder to measure and generally a bit more messy than the other hormones. It's also harder to change because it's further down the gut. But if you look at the figure down in the bottom corner, you can see again we get a stimulation for both the gastric and duodenal treatments, and the duodenal treatment is lasting longer. So it's got that lastability the gastric treatment doesn't. What about insulin? So again, we see a normal insulin response to the breakfast, as you'd expect, goes down, and then to the ad libitum lunch, we're seeing a much greater insulin spike in the placebo treatment, so when they've just taken a placebo pill. And so this is likely a result of both the GLP-1 action, which can not only stimulate insulin, but also improve insulin activity, and also the delay in gastric emptying we'll be seeing from GLP-1 and CCK secretion. So a big decrease in insulin. We also showed an acute change in glucose. So at that immediate post-meal time point, the glucose was lower, but because these are glucose normal individuals, they just normalized themselves after that. So it wasn't significant for the whole treatment effect. And so the big question now is how much did they eat? So did they actually eat less? We've got all these appetite suppressing hormones going to the brain. Did they eat less food? And the answer is yes, they did. So we have reduction of food intake. And so it was lower at the snack, but not significantly, at the lunch, but not significantly lower. But then we had significant decreases in food intake for the snack in the duodenal group, and for the total intake over the snack and the lunch for both of the treatment groups. Right? So again, the duodenal seems to be a bit more effective towards the end of the day, so we're getting that lastability in it. Now, you may be asking, which treatment do we think is more suitable, given they both kind of worked? Well, they both kind of worked, but the gastric treatment was pretty rough. So it was a big hit. So if you remember the figures, right after that lunch, that effect on CCK and GLP-1 was more. And when you look at how these hormones work, at a low level they have no effect, at a higher level they suppress appetite, and at a higher level you get GI side effects. So the gastric treatment was pushing into that GI side effect because it was having a more potent acute effect, but a worse long-term effect. And so we really started seeing nausea events and loose sort of a flushing effect primarily as well as some general gastric upsets on the gastric treatment. Whereas the duodenal treatment, there was one person who experienced nausea and loose stools. So the side effect profile was much more suitable for the duodenal delivery, so that's what we decided to go forward with. Okay, what about using as part of a diet strategy? So that was great. So we've got this, our mechanisms figured out, but that's a model diet. It's not what people do. No one sits there and has this, lunch and then 2 hours later all fixed like that. So we wanted to use it as a diet and a diet strategy. We wanted one where you'd get hunger and food cravings. So where people were really properly hungry. And so we chose a 24-hour water-only fast design. So what we did is we got people there were two studies run here. One was on females and one was on males. We got them to fast for 16 hours at home, unsupervised. So basically they started at 6 p.m. At night, and then they came into the lab at 10 o'clock in the morning, 16 hours of water-only fasting, and then we monitored them for the last 8 hours. So that's the time when a normal individual will really struggle will start getting their hunger increasing. We gave them a treatment capsule so the hop extract at 16 hours and 20 hours into the fast, so that's at 10 o'clock and 2 o'clock, and we measured appetite measures all the way through. So we were asking them, how hungry are you feeling? How full are you feeling? Questions like that. So the first study was done in males with 30 males took part. All of them completed, and the second study was done in 30 females with 27 completing, one withdrawing post-placebo treatment for side effects. So again, that's just they couldn't handle the 24-hour fast, and 2 were unable to complete the study due to scheduling. So this is a little bit of a case study on why a lot of research is not done in females, and this was really challenging. So you have to, for guys, you go, okay, you're coming in on a Monday, and the only thing you have to standardize is they have to be Monday next week and Monday the week after, because you want the same day of the week, because their dietary patterns around the study day might change their hunger. So if you fix it. But for females, you have to get them on the same day of the week and the same time in the menstrual cycle, because menstrual cycle has such a huge effect on hunger and food intake and food cravings. And so it was basically a month to do the study in guys and like 9 months to do the study in females. Yeah. But the results are better in females. So this is the question, how hungry do you feel? And what you see is, so the last 8 hours of the 24-hour fast, you have a marker at the top that says, how hungry are you? And then they just put a line where they're feeling. So I'm not hungry at all on one side, or I'm as hungry as I've ever been on the other, and there's mark it as you go. Now, for the males, what you see is sort of, it's a little bit more jagged. And the reason for it being more jagged is that the guys don't pay very much attention to the questions being asked. So what happens is you'll have a whole bunch of them, and it'll say like, how hungry are you? How full are you? You know, are you having side effects? And they go, yeah. And so you end up with these results where an individual guy will be like, he's getting more hungry, he's getting more hungry, he's not hungry at all, he's getting more hungry. And you know he just hasn't read the question, but you're stuck with the results. The females don't do that at all. They're super careful and the results are beautiful. The other thing that's interesting is that the guys, we get this like, if you look at the male one, at the 90-minute to 120-minute time point, now that's about when they have lunch, so that's 12 o'clock during the day, the guys spike. And the likely reason for that is because a lot of the participants in this study were students who were in a hall of residence, and they had a fixed meal time of 12 o'clock where they were provided food. So they were habituated to have a hunger response at that time. So they would be getting a hormone, or the pro-appetite ghrelin hormone release. So they spiked in the placebo group just then. So, and what's really interesting is both of the treatments with the hop extract, either the high or the low concentration, blunted that response. So they knocked off that increased hunger during mealtime. I think that's a really interesting result for when people are fasting, because that's obviously a time when they're going to struggle when they're thinking about eating. For the females who were more free living, we had just a gentle increase around lunchtime, flattened off a gentle increase around 3 o'clock when they'd snack, and more of a dose response. But for both of the groups, we had really significant changes in hunger profiles that would be expected to result in behavioural change. We gave the females an ad libitum meal. So we gave the males a 24-hour food recall, so we were looking for food 24-hour post-meal, but they basically didn't do it for the last treatment day because they'd had their participant payment and they just left, so we couldn't really use it. So that was a lesson. But for the females, we actually gave them an eat till their full meal, so we'd really, assess how much food they had post-fast. Here's the fullness. So again, the same sort of question: how full do you feel? I'm not full at all till I'm totally full, and the males get a decrease in fullness, but it's important to remember that these people are already quite empty. They haven't eaten for 16 hours, so they're already going to be feeling that they're not very full. And so there's a decrease in the males and the females. It's statistically significant and probably meaningful in the males, but probably not so much in the females. They're probably feeling pretty empty anyway. So a decrease in hunger, but not really a change in how full they're feeling. Now, in the female study, we also asked how much they're craving food. And we thought this was quite an important question to add in, because we use hunger as a marker of sort of likely diet compliance, right? But the really reality is that what you want is it's not so much if people are, needing to eat. People need to eat, that's the way it goes. But if they're craving for unhealthy foods, then they're more likely to make that unhealthy food choice, and you really don't want that. So we asked them, how much are you craving? And the results here are way more potent than for the hunger. And so we see that both treatment groups, they're actually, the craving food goes down, and they're actually craving food less at the 24-hour time point than they were at the 16-hour time point. So they've got less food cravings. So that was really quite amazing. We also showed that when they had the breaking fast meal, that there was a 14% lower food intake. So again, that lines up with the male study, which was an 18% food intake reduction during the mode of action study, where we gave them those ad libitum meals and took the blood samples. In this fasting study, the fast breaking meal was 14% lower. So again, about what you'd expect. So that worked really well. So we got our clinical evidence, and we think this is pretty good. So what about using this kind of product in a clinical environment? Now, there's a little proviso here. I am not a medical doctor, I'm a scientist. So these are suggestions from a science point of view. So the general information about this hops or hops-based product like this is that it's got an active dose of 100 milligrams to 500 milligrams from the studies. So as low as 100 milligrams was shown to work in the female study, in the male study, and then up to 500 milligrams was shown to be highly effective in the first mode of action study. Now there is a potential for some gastric upsets, so we didn't see many. So in the first study we had that one guy who got basically nausea and flushing. In the male fasting study we had in the high dose treatment 3 guys who had flushing without nausea. And in the female study, we had two females who had flushing, again, gastric flushing without nausea. So there are side effects, but they're not very common, and this represents probably a worst case scenario because these people they're treatment naive, so they've not had any chance to adjust. So we recommend probably a dose escalation, particularly if they're getting some side effects. So just dose it up over a few days. So going from, say, 100 milligrams or 125 milligrams up to the 500 they might need. The other consideration is this is gut targeted, and gut targeting is wonderful for a lot of reasons. It means that you don't have to get into the blood of people, which means you don't have to worry about drug interactions and organ issues, interactions with organs. The downside of gut targeting is that it does introduce a little bit more variation between people because you're not concentrating into a fixed volume of blood. You're reliant on how good their stomach is at emptying and how much it disperses, how big their intestine is. It's more variable. So there is a little bit more, I guess, optimization of dose and timing that's required for this relative to other types of supplementation. The mode of action is GLP-1, CCK, and PYY stimulation. Again, this is really good that there's all 3 of these hormones triggered because when you think about that side effect profile, if you get an individual hormone and you start pushing it really high, and you start to get those gastric side effects. And I think that what we're seeing in the pharmaceutical industry is a realization of that, where the new treatments coming out are combinations of multiple hormones, and that when they show lower side effect profiles because of that. And so we're just fortunate we managed to get 3 of them, without having to make a drug. So you get around 4 hours of activation per dose of this product. So some people will see more, some people will possibly see less, but usually it's 4 hours plus. Occasionally you'll get a person who will take 1 pill and they will say, I'm good for a whole day. Yeah, so they'll get a really long-lasting effect. And it also is important to note that it mimics the natural cycles of your hormones. So your hormone goes up when you have a meal. This product just boosts that increase. So it just goes up higher, right? It's not like a pharmaceutical, which is just saturating your system 24 hours a day, 7 days a week. So you get a chance to reset. You do need to give it approximately an hour or so before a meal and on an empty stomach for it to get to the right part of the gut. Animal studies show that bioavailability is low, so low amounts get into the blood, and that the primary point of metabolism is in the colon. Again, that's all good news for drug interactions and safety profiles. Okay, so what sort of patients might use this? Well, anyone who's struggling with hunger and hunger-related conditions, right? So if you've got, either you're trying to improve your diet and you can't because you're constantly eating foods you crave, junk foods, or you're trying to lose weight and you improve your diet through that. So anyone who's got those hunger and food craving conditions. If you're adverse to injections, so if you don't want to inject yourself every week with a GLP agonist, if you can't afford a GLP agonist, or if the side effects are too strict, or too nasty. So there have been a lot of, so again, I'm not a medical doctor, but this sort of product, so this hop extract has been used by medical doctors, and they're using it in a couple of ways. One is essentially a pre-treatment before going on a GLP-1 agonist to say, do you really need to use an agonist? Can you use a natural GLP-1 stimulator and see if that effect is good enough? Some are also using it as a co-treatment, so saying, all right, instead of using a full dose of your GLP-1 agonist, let's give you a smaller injection and we'll give you this alongside, so it brings the cost down and maintains the efficacy. And they're also using it as a safety net when you're coming off the treatments. Because what we know from the studies is that the weight gain or the weight loss that you get from a GLP-1 therapeutic, when you go off it, it comes back, and it comes back pretty much completely within 4 years, and that's complete intervention failure. Yeah. And there's also, I guess, the people who are concerned that those GLP-1 agonists are not acting sort of naturally at all. Even though it's a modified hormone from your body, it's not mimicking your physiological system. Okay, So what are the side effects and contraindications? All right, so loose stools is the most common thing we get, usually not without nausea. So it sort of comes across as oh, I think I need to go to the bathroom now, and it's a loose stool, but you don't normally feel unwell. We occasionally get gastric upsets, and this is likely to improper delivery, where the pill has opened completely in the stomach rather than going to the small intestine. So there's a lot of acid getting secreted into the stomach, causing heartburn, etc. And so if that happens, we're looking at changing timing of treatment, maybe giving it, making sure the stomach's empty, or making sure giving it slightly longer before a meal to get through into the small intestine. If someone's got inflammatory bowel disease, all right, this is probably not the best product to give them because it is gut targeted. It does rely on you having a healthy, or at least a relatively healthy gut to work. I mean, it may well be effective, but again, probably not the best. And if you've got IBS and it's diarrhea predominant, because if you're going to get a side effect, it's going to be flushing or acute stool or diarrhea. If you're already suffering from diarrhea, it's probably not the best. But if you've got constipation predominant IBD, that's fine. Cancers of the GI tract. So particularly if you have an enteroendocrine cell cancer where your hormone levels are already altered, then you're just going to alter them more and stimulate those hormones more, and so you probably don't want that. And if you've got gastroparesis. So this will delay gastric emptying. Right? It won't do it for days. It'll do it for a series of hours. But if you're suffering from gastroparesis, you don't want that. And also if you've had a previous gut resectioning. So this is a case-by-case assessment because it will alter your sensitivity. So if you've had a bypass surgery, then you're going into a more sensitive part of the gut, and so you may have more acute effects, so dosage may need to be tweaked, etc. We currently only have anecdotal evidence for weight loss. So we've got a whole bunch of acute trials that show really promising effects. We know that GLP-1 long-term, if it's stimulated, will lead to weight loss, but we're currently running a weight loss trial now. So right now, as I said, we're doing a weight loss, so we've got the ethics we're recruiting, and it's a 6-month long with a 3-month follow-up, non-treatment follow-up to see what happens post-treatment withdrawal. 75 participants per group, so a placebo group and an active treatment group with diet and exercise advice, and then we give the extract on top of that to see if it helps them maintain their diet. And we also have plans for a post-bariatric surgery trial to see how that works for weight loss post-bariatric surgery. So that's me. So thanks, guys. Any questions? Oh, we've only started recruiting, so we haven't got any preliminary weight loss results. I mean, anecdotally, the data is really good, but the proof's in the pudding for the weight loss. So that'll probably come out towards the end of the year for the preliminary results. Yeah.
Hi, thank you so much. I'm sorry if you already mentioned this, but do you know if there's any medication interactions?
And I guess it's relatively unlikely because there's only a small amount of absorption into the blood, so it's limited gut absorption. So if you were to be concerned, I'd be concerned if there was some sort of medication which was gut targeted is the only time. I mean, I can't think of any off the top of my head, but if there was one, that's when I'd be thinking about, well, at least timing the medication differently. So if you're taking the medication in the evening before you go to bed, if you time the treatment, several hours before that, then that should hopefully resolve that issue.
Hi, thank you for the presentation. Since it's considered as a supplement, do you know who make the decision to prescribe them, or like if there's complications, how do we address that?
Yeah, so that's a good question. So I mean, as a dietary supplement, there's no regulations around who can give it, so anyone can essentially sell it. I think that my preference is that it's managed through medical professionals, and if if there are side effects, that they're discussed between the patient and the medical professional, and they're managed that way. So there's to be honest, there's more information about this in the exhibit hall. But there is, I guess, a support system for practitioners around what to do if a patient happens to get a side effect, and how to address it, and what the system is to, say, alter the dosage, alter the food timing, etc. But this has been available to consumers since 2018, and there's, as far as serious side effects go, there's not been any reported as far as I'm aware.
Thank you for the presentation. It was fascinating. I'm just curious, do you have any handle or idea on the incidence of diarrhea in people that are taking it? Like, what's the chance that somebody would have diarrhea?
Yeah. So I mean, in the clinical studies, what we see is between a 5 and 10% rate of that acute stool without nausea. And the reports from, use by the general public, which again, are anecdotal to me, but are less than 5%. And I think the clinical trials represent what's probably a worst case scenario, because they are given, in some cases, a maximum dose without any adaptation period. So again, we're looking in that sort of range of around 5%, which will typically resolve if someone continues to take it for a period of time. Yep.
And then I have one more question. As a scientist, being an expert in this particular nutraceutical, I know you probably haven't had time to study it, but you'd mentioned that people could possibly take a dose of this and a small dose of an injectable GLP-1 at the same time. Do you foresee any, by combining those two, or do you foresee any safety concerns or synergistic effects that would be of concern?
Yeah, so again, I'm a scientist, not a medical practitioner, but I can't see any particular safety effects. I mean, if you're lowering your synthetic GLP-1 and you're boosting up your natural GLP-1 a bit, along with CCK and PYY, then I think that's quite a good way to mitigate some of the potential side effects and reduce the cost. So I can't see any issues with that, no.
Okay, thanks.
I have one quick question and then another one. You said there were 4 candidates at the end of your trials of all these different foods. I was just curious, was bitter melon one that also helped? Because people do report that it seems to help blood sugars, but I'm not sure of the mechanism.
Yeah, so I mean, we looked at bitter melon. We didn't get particularly great results from it. A lot of the issues were around stability. So with the extract we use, what we have is it has a persistence in the epithelial lining. So what that means is that if you were to, say, get the extract and take one of these pills, break it open and put it on your tongue, you won't be able to get rid of it. So you'll get this persistent bitterness for hours and hours. And we believe that really allows us to get that long-term hormone release. So over the course of 4 hours of hormone release, rather than just an acute little snap like that for half an hour. And so a lot of the products that were sort of excluded, or the plant extracts that were excluded, simply didn't have stability in the gut. So they were breaking down too quickly, whereas the hops was persistent through the entire small intestine.
So until you have the results for the weight loss study and can show that as a reason to take it, what are you advertising this for? Is this to lower blood sugar? You had pretty good response with the insulin and the glucose. If it's for that, does it compare to, say, the diabetes drugs that are the gliptins like Januvia? Or is it metabolic syndrome across the board, or I'm sorry?
Yeah. So again, I don't sell anything, and I get no royalties from sales of anything because I work for the government. Yes, I mean, for me, it's diabetes is probably something to look at in future. We don't really have the data at the moment, and a disease, talking about addressing a disease state when you don't have data for that disease state is probably a bit, that's a future option. But right now it's really for any condition where hunger and food cravings is a challenge. So people think about obesity, that's great, and it works for that, but it's also if someone's of normal weight who just wants to improve their diet. I mean, I'm of normal weight, I can eat garbage food, I don't get fat, but my cholesterol goes up, my blood pressure goes up, etc. So there are people who are like myself who would benefit from it to enable dietary change.
I had another question for you. I'm over here.
Oh, yeah.
Hi. What was the duration of the adverse GI side effects that you had reported?
Right, yeah, so it was typically a singular bowel motion, that happened around 2 hours post
2 hours.
taking the pill, yeah. So I mean, it varies a little bit between people, but in the clinical trials, yeah.
Okay, and the nausea, was it similar, like 2
Similar
hours?
for the nausea
Okay.
side effect, yeah. And interestingly, in the study, the male study where we gave 2 pills, it was 2 lots of acute stools, so basically 1 per pill. So yeah.